Slide 1. Introduction to Medical Genetics. Hello everyone, and welcome to our lecture on medical genetics. Genetics, at its core, is about understanding how traits are passed down from parents to offspring. This includes delving into the molecular structure and function of genes, observing how genes behave within cells and organisms, and studying the distribution and variation of genes within populations. Today, we'll be focusing on the application of these principles to medical care. Genetics is a vast field with many specialized branches. While we won't be covering all of them today, it's important to recognize the breadth of the discipline. Some key branches include medical genetics, which is our focus, as well as microbial, molecular, population, archaeogenetics, psychiatric, and quantitative genetics. Each of these branches offers a unique perspective on the role of genes. So, what exactly is medical genetics? It's the branch of medicine dedicated to the diagnosis and management of hereditary disorders. It's a field that bridges scientific research with practical medical applications, using our understanding of genes to improve patient care. Medical genetics involves applying genetic principles to medical practices. This includes studying how diseases are inherited within families, mapping disease genes to specific locations on chromosomes, analyzing the molecular mechanisms by which genes cause diseases, and ultimately diagnosing and treating genetic diseases. Genetic disorders arise from abnormalities in an individual's DNA. These abnormalities can range from small mutations in a single gene to the addition or subtraction of entire chromosomes. Understanding the nature of these abnormalities is crucial for diagnosing and managing genetic diseases. Genetic disorders often share certain characteristics. They can be congenital, meaning present at birth. They also exhibit specific modes of inheritance, follow particular patterns of population distribution, and often have a familial component, meaning they tend to run in families. Genetic disorders can be broadly classified into several categories. These include single-gene disorders, chromosome disorders, multifactorial disorders, somatic cell genetic disorders, and mitochondrial disorders. Each category has distinct underlying mechanisms and patterns of inheritance. Single-gene disorders occur when a mutation alters or eliminates the protein product of a single gene. Examples include cystic fibrosis, Fragile X syndrome, and sickle cell disease. These disorders often follow predictable patterns of inheritance, making them easier to trace within families. Chromosome disorders involve missing, duplicated, or otherwise altered entire chromosomes or large segments of them. Patau syndrome, also known as trisomy 13, is an example where there is an extra copy of chromosome 13 in all cells. These disorders often have significant developmental consequences. Multifactorial disorders result from mutations in multiple genes, often in combination with environmental factors. Common examples include obesity and hypertension. Somatic cell genetic diseases like cancer arise from altered genetic material in somatic cells, which are non-reproductive cells. Mitochondrial genetic disorders affect the mitochondria, the energy-producing parts of our cells. These disorders are often due to mutations in mitochondrial DNA. Examples include mitochondrial myopathies, which cause muscular problems, and Leber hereditary optic neuropathy, LHON, a frequent mitochondrial disorder that causes visual failure.